Video 5 Molecules of Life. Subtopic DNA and RNA molecules. The learning outcomes for this subtopic: first, state the structures of nucleotide as the basic composition of nucleic acid. Second, state the types of RNA. Nucleic acids exist in two different forms called DNA or RNA. DNA refers to deoxyribonucleic acid, while RNA refers to ribonucleic acid. Nucleic acid is a group of complex compounds found in all living cells, such as humans, plants, and bacteria, and also viruses, that is essential for life. They function in controlling cellular function and heredity, for example, for encoding, transmitting, and expressing genetic information. The basic composition of nucleic acid, also known as the monomer of nucleic acid, is called nucleotide. Nucleotide of nucleic acid made up from three different components, which is phosphate group, pentose sugar, and nitrogenous base. There are two types of pentose sugar. First, deoxyribose sugar and ribose sugar. Deoxyribose sugar only can be found in DNA, while for ribose sugar, it can be only found in RNA molecule. For nitrogenous bases, there are two major groups that are differ in their structure. We have purine, consists of double ring structure, and then the second one is pyrimidine, consists of single ring structure. The example for purine nitrogenous base, there are two types, adenine and guanine. For pyrimidine nitrogenous base, there are three examples, which is cytosine, THYMINE in DNA, and URACIL in RNA. This diagram shows the structure for RNA and DNA molecules. Even though both molecules are types of nucleic acid, but they have different structure, in which we can see that for RNA molecule, it has only one or single polynucleotide chain. Polynucleotide chain form when many nucleotides join together. For DNA molecule, it has double polynucleotide chain, 1 and 2. And these two polynucleotide chain join together by hydrogen bonds between nitrogenous base. For example, we have two hydrogen bonds that form between adenine and thymine base, and three hydrogen bonds form between guanine and cytosine base. DNA and RNA molecules also have different types of nitrogenous base, in which for DNA molecules, there are four types of nitrogenous base that can be found, in which we have thymine, represented by capital letter of T, adenine, A, cytosine, C, and guanine, capital letter of G. Meanwhile, for RNA molecules, there are also four different types of nitrogenous base that can be found in RNA, in which we have uracil, represented by U, adenine, cytosine, and guanine. Chargoff rule is a rule that used to figure out how the four bases of DNA match up in pairs. So based on this rule, adenine base always pair with thymine, and guanine always pair with cytosine. This diagram shows how thymine and adenines pair together or join together by two hydrogen bonds. And this diagram shows how cytosine pair with guanine linked together by three hydrogen bonds. As we know that there are two types of pentose sugar. First, deoxyribose sugar found in DNA and ribose sugar found in RNA. Pentose sugar refer to the 5-carbon sugar, carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. Both the structure of deoxyribose and ribose sugar almost similar, but they are differ at the carbon number 2, in which for deoxyribose sugar, carbon number 2 attached to two hydrogen atoms, while for ribose sugar, carbon number 2 attached to hydrogen atom and hydroxyl group, meaning that there is no hydroxyl group attached to the carbon number 2 of deoxyribose sugar of DNA. The next is nucleotide formation. So what is nucleotide? Nucleotide is an organic compound consists of nitrogen containing purine or pyrimidine base, sugar, either ribose or deoxyribose sugar, and then phosphate group. So these three components must combine together in order to form a nucleotide. So how to draw nucleotide? So the three components of a nucleotide consist of pentose sugar, nitrogenous base, and phosphate group, symbolized by capital letter of P. Pentose sugar is a 5-carbon sugar, carbon 1, 2, 3, 4, 5, and this one is oxygen atom. So when you draw nucleotide, make sure nitrogenous base join or attach to the carbon number 1 of a pentose sugar, while phosphate group attached to the carbon number 5 of pentose sugar. There are three types of RNA. First, messenger RNA, mRNA. Second, ribosomal RNA, rRNA. And the third one is transfer RNA, tRNA. All the three types of RNA have specific function, in which for messenger RNA, it copies genetic information from DNA to the ribosome during transcription, and also it serves as template during translation or during protein synthesis. For ribosomal RNA, it assembles or combines with the protein to form ribosomal subunit in the cytoplasm, and then it also involved in protein synthesis. For transfer RNA, it will transfer specific amino acids to the ribosome during translation. This is the general function of mRNA, rRNA, and tRNA. So, genetic information in DNA will be copied into mRNA, and this process occurs during transcription inside the nucleus. mRNA then will be transported to the cytoplasm of the cell, and then here it will serve as a template to form protein. So during formation of protein, specific amino acid will be transferred by tRNA molecule.